On the agenda is a brief overview of the Advanced High Throughput Flow Cytometer, the IQ platform, which was used to create the data seen in this webinar. I will then give an insight into how T-cell exhaustion occurs and how this progresses, specifically with regard to an oncology setting, and how the tumour microenvironment plays a role here. Finally, I will cover two case studies. The first looking at BiTE antibodies and their effect on T-cell exhaustion compared to a more rudimentary activation method. And the second will cover how the T-cell exhaustion kit provided by Sartorius can allow us to identify CAR T-cell activation. The IQ3 is an advanced flow cytometer capable of reading from either 96 or 384 well plates at high speed. The IQ3 also allows a vast quantity of information to be collected from up to 15 detection channels. The optimized assays and reagents provided by Sartorius require a small volume of cells and supernatant and therefore minimize the amount of precious sample required. The IQ3 system comes with built-in Foresight software allowing for fast time to results as graphs auto-populate as cells are being sampled. As I have mentioned, the IQ3 has a suite of compatible reagents that are applicable to a range of different applications, namely within the immuno-oncology, antibody discovery, and foundational areas. The IQ3 allows a user to simultaneously test T-cell phenotype alongside secreted proteins in a 10 microliter sample from a 96 or 384 well plate. This provides insight into the status of multiple cell types within a complex cell environment, such as the quantification of CD3 positive lymphocyte populations, live and dead cell readouts, and also cell marker expression. Q-beads included in the wells allow the capture and quantification of multiple specific protein or cytokine readouts, such as TNF-alpha and granzyme B. I will now begin to discuss how T-cell exhaustion comes about within an oncology setting. T-cells within the tumor microenvironment interact with antigen-presenting cells, or APCs, which present the cells with a foreign antigen. This stimulates the T-cells to activate via the T-cell receptor pathway and/or CD3. Activation of T-cells induces low-level upregulation of the PD-1 and LAG-3 markers to both control the magnitude of the initial activation and also to act as a negative feedback loop. Upon activation, the T-cells produce a number of cytokine and chemokines to stimulate other immune cell types to respond. The activated T-cells also act by killing the cancerous cells. Over time, antigen-presenting cells begin to produce inhibitory signals. Tumor cells also release inhibitory factors into the tumor microenvironment, such as tryptophan. Other cells in the tumor microenvironment, such as tumor-associated fibroblasts, inhibit T-cell function and activation via Fas ligand. Accumulating inhibitory signals and long-term exposure to the same antigen cause increasing inhibitory receptors to be present on the T-cell. These include PD-1, LAG-3, CTLA-4, Fas, TIM-3, and many more. As the tumor begins to escape the immune system and proliferate, mounting inhibitory signals are produced. Increased complexity forms within the tumor microenvironment as increasing inhibitory cell types infiltrate, such as tumor-associated macrophages. These further aid the tumor in its growth. This blocks appropriate T-cell response, such as cytotoxicity, proliferation, and cytokine production. This loss of effector function is otherwise known as T-cell exhaustion. Sartorius have produced the T-cell exhaustion cell and cytokine profiling kit, which is available for violet, blue, red laser IQ and IQ3 systems. This reagent kit allows for the quantification of viability, proliferation, T-cell subtypes CD3, CD4, and CD8, T-cell status markers PD-1, LAG-3, and TIM-3, and also cytokine secretion of interferon gamma and tumor necrosis factor alpha, all from a single well. Cytokine standards are provided to allow quantification of protein production, and predefined compensation settings allow the user to quickly ascertain their results. Also provided with the T-cell exhaustion kit is the predefined gating strategy. This speeds up the time to results as these gates are auto-populated whilst the data is acquired. Foresight also allows the fine-tuning of these gates by using positive and negative populations. Initially, Q-beads and cells are clearly separated using the forward scatter and side scatter graph. Singlet beads and cells are then selected for, and Q-beads are then split into TNF-alpha and interferon gamma based on the bead's internal fluorescence. The single cells can then be split into effector and B green encoded target cells. If target cells are not used, then the effector cell may be encoded with the B green encoder dye provided with the kit. Viability of effector cells can then be deduced, and live effectors can be taken forward into the phenotype gates. CD3 cells are first identified, and these are further phenotyped into CD4 helper and CD8 cytotoxic populations. Finally, PD1, LAG3, and TIM3 positive cells can be identified in each of these groups. To first evaluate T-cell exhaustion, CD3 positive T cells were treated in one of the following three ways. Non-stimulated, shown by the blue histogram peak, stimulated once with Dynabeads CD3, CD28, represented in light grey, or stimulated with Dynabeads repeatedly every 2 to 3 days for a period of 7 days, shown in dark grey. Dynabeads activate T cells via the CD3 T cell receptor pathway and also using a co-stimulatory signal via anti-CD28, mimicking the interaction in vivo with antigen-presenting cells. Here we show a series of histograms of PD1 expression on CD3 positive cells. Gates were set based on the day 0 populations. As you can see, after 2 days, both of the stimulated populations increase in PD1 expression, indicated with a right shift in the histogram. At day 5, we see the single stimulated population decrease, i.e. shift to the left, while the repeat stimulation maintains its PD1 expression. Finally, at day 7, the single stimulation returns back to non-stimulated levels, while the repeat stimulation sees sustained expression of PD1. The same repeat stimulation was used to pre-exhaust CD3 positive cells in this assay. Non-exhausted cells from the same donor were used as a comparison here. The cells were plated in well and treated once on day 0 with a dose response of Dynabeads CD3 to CD28. A 10 microliter sample was taken on 3 sequential days from the same plate and analysed for expression of exhaustion markers and cytokines. On day 1, 60% of exhausted cells express PD1 irrespective of the concentration of Dynabeads used to re-stimulate them. LAG3 positive expression is also very high in exhausted cells, ranging from between 80 to 100%. The non-exhausted cells exhibit low levels of both PD1 and LAG3 on day 1, dependent on the density of Dynabeads they were stimulated with. Day 2 shows a similar pattern with increasing levels of exhaustion markers on non-exhausted cells, while those that were pre-exhausted maintain high and consistent PD1 and LAG3 expression. By day 3, non-exhausted cells match or exceed the exhausted population when looking at both PD1 and LAG3 expression at high densities of Dynabeads. When looking at the full time course, it is clear that PD1 expression decreases slightly in pre-exhausted cells re-stimulated with low densities of Dynabeads. However, this is not the case with LAG3, where expression is sustained at all stimulation concentrations in pre-exhausted cells. Cytokines from the same assay plate were also assessed. Non-exhausted cells show concentration-dependent increase in the production of TNF-alpha from day 1. In contrast, exhausted T cells produce significantly reduced concentrations of TNF-alpha over the time course. The highest bead stimulation, using 200,000 Dynabeads per well, caused non-exhausted cells to produce 12-fold more TNF-alpha than pre-exhausted cells with the same stimulation. TNF-alpha production peaks at 0.7 nanograms per mil when exhausted cells are treated with 12,500 Dynabeads per well. Reduced TNF-alpha production is a hallmark of T-cell exhaustion, and this reduction can easily be detected and quantified alongside marker expression using this kit. I will now move on to how bispecific T-cell engager antibodies function and cover some data as to how this affects the exhaustion phenotype seen. BiTE antibodies are made up of fragments from 2 antibodies attached by a hinge that function by binding to both the T cell and the target cell simultaneously. This brings the activated T cell into direct contact with the specific target cell of interest. In this case study, we've used an anti-CD3 cross CD19 BiTE antibody. This exclusively binds to CD19 positive target cells, such as those in B-cell lymphomas, allowing specific target cell killing. Once the antibody has bound to both target and the T cell undergoes clonal expansion and proliferation, while the target cell is attacked and eventually will undergo apoptosis. This anti-CD3 cross CD19 antibody was used in an immune cell killing assay set up in comparison with a more crude method of T-cell activation, namely Dynabeads CD3-CD28. NUC-like green labelled Ramos target cells were used as our CD19 positive target cell line, and PBMCs isolated from whole blood were used as our effector cells. After 60 hours in plate, a 10 microliter sample was taken and analysed with the T-cell exhaustion kit. As you can see, PD-1 expression is concentration dependent with both activator types, but is expressed at a higher level in Dynabeads compared to the CD3-CD19 BiTE. This can also be reviewed directly from Foresight software in the form of a heat map shown on the right. LAG3 also follows this trend, with higher expression seen in PBMCs activated with Dynabeads compared to BiTE. At highest bead densities, we see 25% of CD3 cells expressing LAG3, compared to just 12% when treated with the highest concentration of BiTE, 10 nanograms per ml. TIM3 expression is relatively low across all treatment types and highlights the temporal nature of these exhaustion markers, whereby PD-1 is expressed shortly after activation, followed by LAG3, and finally TIM3 after high levels of stimulation to the same antigen. When looking overall at the levels of exhaustion markers, one might assume that because the exhaustion levels are higher in the Dynabead wells, that more killing will have occurred here. However, the count of live target cells was also collected from the T-cell exhaustion cell and cytokine kit by using the NUC-like green labelled Ramos cells. These can easily be separated from the effector cells using the BL1 versus side scatter height dot plot as shown on the right. As you can see, at highest concentrations of BiTE, we see a 31% decrease in the number of target cells compared to the highest Dynabead density. The insight achieved from multiple aspects of the kit allows for the identification of more specific activators that may enhance our ability to tackle specific tumour types in vivo. This case study highlights how the BiTE antibody induces lower PD-1 and LAG3 expression after 60 hours in co-culture, showing the more specific and gentle activation approach causes lower levels of exhaustion despite enhanced killing of specific target cells when compared to Dynabeads. The final case study utilises anti-CD19 chimeric antigen receptor T cells, or CAR T cells, to target a CD19 positive B-cell lymphoma. These CAR T cells have been engineered to specifically identify CD19 on a target cell. When this occurs, the T cell will activate and undergo rapid clonal expansion and attack the CD19 positive cell, causing tumour cell death. However, upon interaction with a CD19 negative cell, no such proliferation nor attack should occur. This makes these cells highly specific and useful in a clinical setting to target cancerous cells, while leaving antigen-negative healthy cells untouched. We compared CAR T cells to non-activated and Dynabead-activated T cells obtained from the same donor over the course of 3 days. The CAR T cells used are 54% antigen positive, meaning 46% of cells within the CAR T wells are normal, non-transfected T cells. Effector cells were plated in the presence of CD19 positive Ramos nut green cells or CD19 negative Yurkat nut green cells. 10 microliter samples were taken on 3 sequential days and analysed using the T-cell exhaustion kit. When looking at TNF-alpha and interferon gamma production, it is clear that across the 3 days, there was little response when non-activated T cells were present. T cells with activation, however, saw increased cytokines over the 3 days. Interestingly, this was higher in the presence of Ramos cells than Yurkat cells in both cytokine readouts. When looking at the CAR T cell response, we see high levels of cytokine production, but only in the presence of the CD19 positive target cell line, showing that the CAR T cells are responding and activating in response to these cells. Furthermore, we utilised the multiple metrics obtained by the Multiplex IQ kit to assess effector cell proliferation and target cell count. Non-activated effectors do not exhibit much proliferation over the 3 days, and the same is true for the activated T cells. Target cell proliferation is also apparent in wells with non-activated cells. However, when activated T cells are included, we see enhanced killing of Yurkat cells. As this is preliminary data, we are still looking to complete further studies to elucidate why this is occurring. When looking at the CAR-T data, we see significant CAR-T proliferation in the presence of the Ramos cell line, again suggesting the CAR-T cells are activating in response to the CD19 positive stimuli. Corresponding decline in Ramos cells compared to the non-activated control is also seen. These results further identify the enhanced activation that CAR-T cells are undergoing in the presence of their target cell. The expression of PD-1, LAG3, and TIM3 was also collected. However, due to the early time points of this dataset, we are showing PD-1 data as an early marker of activation. In non-activated T cells, we again see little to no PD-1 expression across the 3-day time period, while activated T cells show maintained PD-1 expression around 10% in the presence of both target cells. Again, we see a difference when CAR-T cell effectors are present, with increased PD-1 expression in the presence of Ramos cells when compared to low, non-activated levels of PD-1 in the presence of CD19 negative Yurcats. In summary, TNF-alpha and interferon gamma cytokine release shows the CAR-T cells are activating in response to the CD19 antigen-positive cell line. CAR-T cell proliferation is higher in wells with Ramos cells compared to CD19 Yurcat cells. A decrease in total viable CD19 positive target cells can be seen, highlighting the specific killing of the Ramos cells. Early exhaustion marker PD-1 is also more highly expressed when Ramos cells are present. This case study helps to identify an alternative use of the T-cell exhaustion kit, highlighting that it can be used to first identify activation of effector cells, before later time points identify the exhaustion of cells by decreased cytokines and increased late exhaustion marker TIM3. To summarize, the IQ Advanced Flow Cytometry platform and its compatible reagents allows the simultaneous detection of cells and cytokines from a single 10 microliter sample per well. Utilizing the human T-cell exhaustion cell and cytokine profiling kit allows successful identification of exhausted cell phenotypes, which can be tracked over a period of days to allow a timeline of events to be elucidated. Therapeutics or gene-edited cells of interest can be used in the presence of a target cell type. This allows greater insight into the specificity and function of these potential treatment types.